I work at Vanderbilt University in Nashville, Tennessee, and I'm interested in neurodegenerative diseases such as Parkinson's disease. And most of the work that we do is looking at interaction between genetics and the disease using various environmental agents. The ones that we are most interested in are different metals, such as mercury and manganese, especially manganese because it has been shown to have symptoms that are very similar to Parkinson's disease. So we use different models for our experiments. One that we use quite often is working with little worms, and they're very contemporary genetic tools that can be used to mark specific cells by using different genes. So we can very easily identify which of the cells are specifically affected by the metals. And then we can manipulate the expression of the genes and see how we can rescue the cells, how we can make things worse, and determine the mechanisms by which the different metals affect the viability and the function of these cells. So I was approached to do some work in the model that we have with deuterium-depleted water. I'm by no means an expert in deuterium-depleted water, and I was actually very skeptical when we first started the work. But given the fact that we have this model where we can look at the viability of the cells, the viability of the worms, we decided to see whether it's going to work. So the model that we used is the same model, namely we used the worms, and we looked at aging in the worms and different pathways that are known to be important in regulating the aging in the worms. So it's a very simple experiment where we treat the worms with in their diet they also get the deuterium-depleted water, and then we can look at their survival. We can look at the expression of different proteins that are involved in regulating aging processes. And this was all done within the context of a model that we know has a shortened lifespan, and for that we used the manganese. And I was really surprised basically because we have been shown that if we take animals that are, for example, treated with manganese, we can reverse many of the effects that we see with manganese. And the bottom line really is that the animals that get the manganese and the deuterium-depleted water actually live the same length as do the animals that serve as control. So we see this complete reversal of the effects of manganese. Most of the work that's been done has been really on cancer. I have not worked in the cancer area. I think one would obviously need to do, with the cancer data that we've heard over the last couple of days, one would have to do a phase 3 study, which is a lot more expensive, takes a lot of time, and it has to be double-blinded and randomized kind of study. But I think once that kind of study is done, it will give a much more definitive answer on the ability of the product to change the outcome of the disease, prolong life, do all kinds of things that are good for humans in general.